The mitochondrion is the powerhouse. Powerhouse?
Powerhouse! Powerhouse of the cell!
As a biology teacher, I know a fair amount of what scientists mean when they say that the mitochondrion is the powerhouse of the cell. But recently I realized that I know almost nothing about how we know that that is true. Something so widely repeated should have an iconic story, right? Like Newton and gravity or Darwin and evolution. So I decided to find out what that story was. But what I found did not fit the usual tropes of scientific discovery stories. Here is what I learned. Our story begins with a mystery that people were wrestling with all the way back in the 1700s. We knew that life required energy to move, to mate, to grow. But where does that energy come from? What powers life itself? In 1783, the father of modern chemistry, the French scientist Antoine Lavoisier, posited that there must be some kind of internal fire burning inside of all of us. After all, he reasoned, we all consume fuel, give off heat, take in oxygen, and give off carbon dioxide as an exhaust, just like a fire. But in science, ideas need to be tested. And to test Lavoisier's idea, we would need to know where this fire was happening, how it was burning, and what was controlling it. To solve this mystery, just like in the board game Clue, you would have to find the right room, the right weapon, and the right suspect. Now, burning is a chemical reaction. And if something was burning inside the bodies of living things, that reaction had to take place somewhere. But where? What was the room? Was there some kind of internal fire organ? Lavoisier thought it was the lungs. Other scientists thought it was the blood, but really it was all just speculation. And for nearly a century, speculation was all we had. It took a series of truly Frankenstein-esque experiments to settle the issue, in which scientists took various body parts out of the body, tried to keep them alive, and see if they produced carbon dioxide on their own. In one particularly gruesome example, a German physiologist named Edward Pflüger cut off a frog's head, kept its body alive, and replaced its blood with salt water. When the amount of carbon dioxide that the frog's bloodless body dropped by only a little bit, Pflüger concluded that it could not be the blood alone. As these experiments continued, it became clear. It wasn't the lungs. It wasn't the blood. It was everywhere. Every organ, every cell in your skin and your heart and your bones. Which room was the crime committed in? It was all of them. So we'd figured out the room. But what was the weapon? What was the mechanism for this burning happening in all our cells? Straight combustion would release all the energy from food at once, so fast that the cells wouldn't be able to make use of it. It'd be like trying to run a car by lighting the gasoline with a match. Cells had to be controlling this burning somehow. A possible answer came in the late 1800s with the discovery of enzymes. Enzymes are complex molecules that can control chemical reactions by literally grabbing other molecules, taking them apart, and putting them back together again. And chemists at this time were finding that cells were full of enzymes. The more they looked, the more they found. By feeding different chemicals into enzyme solutions, chemists could figure out what these enzymes did. And by the late 1930s, a complicated precision chemical pathway had been figured out. There were dozens of steps, each controlled by its own enzyme, releasing the energy from food bit by bit by bit. There wasn't one murder weapon here. There was an arsenal. Complicated, precision, molecular pathways that boggled the mind. Our internal fire wasn't a single furnace. It was an assembly line. But even knowing that assembly line in great detail, we still couldn't tell what part of the cell the factory was in. So where are we at? By the 1940s, scientists had figured out the room and the weapons. What about the suspects? By this point, scientists had found all kinds of little cell parts. They called them organelles, but they really had no idea what most of them were for. Were any of them the site of Lavoisier's internal fire? Was it a combination of several of them? Or maybe it didn't happen in any of them at all and just took place in the fluid inside the cell, the cytoplasm. The only way to catch our suspects red-handed would be to somehow get a functioning batch of all the same kind of organelle together. But how do you do that with something so small and without, destroying it? Then, in the 1930s, a Belgian scientist working in New York by the name of Albert Claude figured out a way. He ground up cells to break their membranes and then filtered those membranes out until he was left with a fluid that was basically like cell guts, just the cytoplasm with the organelles in it. Then he put this fluid in a container and spun it in what was basically the high-speed version of the spin cycle on a washing machine, a machine that we call a centrifuge. Now, in the centrifuge, the heaviest bits are flung out to the side. By siphoning off these outermost heaviest bits and then spinning the leftovers again at ever-increasing speeds, Claude could separate out organelles by weight, heaviest to lightest. With Claude's new method, cell biologists could look through microscopes at each new batch to figure out which organelles were in them, and the biochemists could add chemicals to each batch to figure out which enzymes were in them. And of all the different samples, one, and only one, contained nearly the entire arsenal of enzymes. It was the one full of mitochondria. Our suspect was holding all the weapons. The whole assembly line was in one place, an energy factory, a powerhouse. The mitochondrion is the powerhouse of the cell. The simple idea in these 8 words wasn't discovered in some stroke of genius. But I think this kind of story is just as inspiring. Look at how people cooperated. It took centuries of effort by hundreds of scientists in dozens of different countries. Regardless of borders or origins or personal jealousies, we shared information with each other in search of a truth. We don't do science alone. We do it together. It really is one of the greatest group projects in the entire history of humankind.